would like to welcome you to the Best for Last lecture here at the College of Science and Health Symposium series. And everybody knows that biology is the best. That's what
The pressure is on.
There you go. And so I'd like to introduce to you today Dr. Joseph Wilson, goes by Joe. And he is actually a former UVU student. He graduated from here. So, yeah, exactly, go Wolverines, right? So that's one of the major joys of having him coming in and talking to us, because we want you guys to see what kinds of things, that are out there for you in the future. So he graduated from here. He went on to get a PhD at Utah State University, doing work on wasps, so these velvet ants, which are, as he'll talk about more, very wasp, what would you say, equivalent almost.
He's a waspy guy.
There you go. So the point is that he's been into stinging things for quite a long period of time. And if you've noticed that there's some concern now about these gigantic Asian wasps and whether they're starting to take over. So he'll be our first line of defense. He'll be Asian wasp free, no doubt. So he went there. He also did a postdoc in Reno, but he didn't gamble. So he's a good guy about that. But I want you to appreciate that after he did that postdoc, you will find a lot of times that when students go to a university, that the university, when they recognize somebody really good, they know that and they want to keep them. So Utah State University basically asked him to come in, and now he's teaching for them at Tooele and doing a lot of work again on stinging things, velvet ants, and that's why he's here today to talk to us about the evolution of color mimicry in velvet ants. And so with that, I'd like to turn it over to Dr. Wilson.
Thank you. I want to see if I'm blocking the screen so where I can stand. So, Utah State University, Tooele. Did anyone know there was a Utah State University in Tooele? Yes. I didn't know that until I got the job there. So, you're ahead of me. But it's a nice place. Tooele is nice. If you've never been there, you should visit for a while. So, evolution of color mimicry in North American velvet ants. Here's what I'm going to talk about today. I outlined it so we can try to keep me on track. First, what is mimicry? I assume that we're at different levels of our remembering of mimicry stuff, so we'll talk a little bit about that. Then what is a velvet ant? Because most people, including entomologists, aren't very familiar with velvet ants. And then I'll talk about mimicry in velvet ants, specifically within velvet ants, then velvet ants and other wasps, and then something called imperfect mimicry, which I'll explain in a second. But first, mimicry, a brief definition of mimicry. You can probably find this on Wikipedia. But mimicry is the similarity of one species to another, resulting in a benefit for one or both species. So here's an example. Who is that? That's Elvis, right? Well, who's that? That's Elvis. So in fact, that is a mimic, and that is the model. So how does this mimic get a benefit from looking like that other species? Well, he gets paid by looking like that guy. So we have a mimic and a model. And in some mimicry, that's the idea. So in biology, I forgot. So when you have a group of mimics together, like this, they're all mimicking that same model. We call this a mimicry ring. Oops. Or a mimicry complex. In a mimicry complex, there's variation in how well the mimic matches the model. And you can see in here, some of these match Elvis pretty well, some of them don't match as well, but there's also a lot of imperfect mimicry. So these are mimics of Elvis, and there's a variety of imperfectness involved with that. And so sometimes the mimic doesn't exactly match, but apparently these mimics are still getting a benefit by looking like that model. And so there still have to be some benefit involved, but we have imperfect mimicry. So in biology, we have mostly separated mimicry into two groups, Batesian mimicry and Mullerian mimicry. So first, Batesian mimicry is when we have a palatable, it's a tasty or harmless mimic that resembles an unpalatable or a harmful species. So butterflies are often examples of this. So we have three different species that taste bad, 3 butterflies that taste bad. And we have one butterfly species that has 3 different color morphs and it tastes fine. It looks like bad tasting ones when their ranges overlap. So then a bird sees this one and thinks, oh, I'm not going to taste that because I know that they taste bad. Another example for the non-entomology people here are poison dart frogs. As their name indicates, they're poison. And then we have some harmless frogs that look a lot like them. So a snake will see this one and probably decide not to eat it because it knows that's bad. Other example, coral snake versus milk snake. That's the whole rhyme. If red touches yellow, you're a dead fellow. And the red touches black, something about Jack. You're okay with Jack. You're a friend of Jack. So anyway, if red touches yellow, it's bad. More bait-seed mimics. These ones I like more because there's stinging stuff involved. We have a honeybee. Many of you know that they sting. And then we have all these other insects that kind of mimic honeybees. We have stingless flies, so these are flies. That's a moth, and then these are beetles on that side. A lot of bait-seed mimics. So remember, bait-seed is something that's harmless, but it looks like something that's bad. The other kind of mimicry is called Mullerian mimicry. It's when two or more species share a color pattern for mutual benefit. So this is a classic example. Monarch butterfly, viceroy butterfly. The monarch tastes bad. If you're a bird, you eat it and it makes you want to throw up, or you do throw up. Viceroy, recently discovered, also tastes bad. So if a bird eats a monarch and figures out it's bad, it's going to avoid everything that looks like a monarch. So the viceroy benefits. If a bird eats a viceroy, it tastes bad, and so then the monarch benefits from it. So they're both tasting bad, but they benefit by training the predator in one step rather than two. Examples of this that are often seen in the textbooks are these butterflies in the tropics. These are heliconius butterflies. We have 4 species, 3 on the top, and this is a variable species. They all look like each other, well, in various ways, and they're all toxic, so Mullerian mimicry. In the Appalachian Mountains here in North America, we have millipedes that 4 different species, they resemble each other. They're toxic also, so nothing wants to eat them because they all look bad. What about velvet ants, though? Before I can talk about mimicry and velvet ants, what is a velvet ant? So velvet ants are actually wasps. They're wingless wasps. Solitary wingless wasps. There's no velvet ant hive, there's no velvet ant queen. They're just solitary wasps, live by themselves. They're sexually dimorphic. That means the sexes look different. These 3 at the top are females. This one on the bottom is a male. If you might notice, one of the big differences between them is the males have wings, females do not. Often the males and females are also different colors. What do velvet ants do? Well, we don't know a lot about the ecology or natural history of velvet ants, but as far as we know, they parasitize the larvae of bees and wasps. So they'll find a solitary bee nest in the ground, and they'll go in there and lay their egg that will kill the baby bee and parasitize it. Velvet ants are really diverse, especially in North America. There's over 600 species. About half of those species are nocturnal, and that's what I worked on for my dissertation work. But the other half are these pretty diurnal-looking ones. Velvet ants are also well defended. Unlike a lot of the Mullerian mimics that we talk about that just taste bad, these ones do more than just taste bad. And this aposematic coloration is a warning coloration. This orange and yellow, orange and black or red and black patterns is often a warning color in nature. So what are these warning predators about? Well, first and foremost, they have a painful sting. You can see how long the sting is on that. It's about as long as the abdomen. And so, unlike a yellow jacket or a honeybee, they can really twist their sting around and sting pretty much whatever grabs them. And I've never been stung by one. I tried to avoid it, but I hear it hurts really bad. They also do this thing called stridulation, which is squeaking. And if I was a fancy PowerPoint maker, I would show you, but instead I just did that. They squeak. So they rub their abdominal segments together and make a noise. They also have a really strong cuticle. Their shell, so to speak, is really hard. And so what all these things put together means is if I'm a predator, let's say I'm a lizard, I bite a velvet ant, it's too hard for me to crush with my mouth because of the strong cuticle. It stings me in my mouth, so I let it go, and then it squeaks as it runs away. So I see its bright colors and I hear its squeaking sound, and I learn really quickly, I'm not going to try to taste that again. And so velvet ants are well defended, but again, what about mimicry in velvet ants? So with 600 species, we have to decide something. Where do we start? How do we investigate mimicry in velvet ants? So I chose this genus, Dasymatilla. These are all members of that genus. It's the most diverse genus of velvet ants in the United States. And it's important, or it's a good candidate to study because the species are often diagnosed by color. So people say this is one species because it's orange or black, this is another species because it's white-ish. So I asked the question, does Mullerian mimicry occur in Dasymatilla? And I've identified 3 characteristics of Mullerian mimics that I can use to answer this question. So in Mullerian mimicry complexes, we should see color patterns that, 1, occur in multiple species. So they need to be shared color patterns. 2, it needs to evolve from convergence, not from common ancestry. So I look like my brother, but it's not because I'm mimicking my brother or he's mimicking me. We look like each other because we share common ancestry. So in order for it to be Mullerian mimicry, it needs to evolve from convergence, which means 2 distantly related or unrelated things look like each other. Then the last one is they need to co-occur geographically. If you're a velvet ant in Utah and you look like a velvet ant in China, that's not going to train any predators to avoid you because there's not very many predators moving from China to Utah regularly. So the first of these 3 characteristics. Do color patterns occur in multiple species? So we have a bunch of Dasymatilla species here. If we just circle a couple of them, we can see, well, that's an obvious color pattern. It's in 3 different species. We have this color pattern that's kind of a grayish, yellowish. It's in multiple species. We have this spotted one that's in multiple species. So yes, color patterns occur in multiple species. And I expanded that search to look at 67 species of Dasymatilla and identified 6 distinct patterns, as you can see here, 6 different color patterns, with at least 7 species in each one of these color patterns. And you'll see this figure again and again, but you can see that those kind of look like each other, and so do those, and so do those, and so on. But you can just look at that and say, yeah, they look like each other, but I wanted to be a little bit more rigorous, use some more statistically based analyses to see if they look like each other. So what I did is I measured different characteristics of all these specimens, how long the hair was, the color and spots and all these different characteristics, put that into a computer program, did a non-metric multi-dimensional scaling analysis, and there's a plot that resulted from that analysis. So what this does is that it plots out each dot there is one of these species, or one of these specimens, and then I can ask if those group together statistically, or if they're significantly grouping together. So we can see like these black ones are these ones right here, and yes, they group together, so they look more like each other than they do like other things. Or these blue ones, which represent those, they group together more than they look like other things. And so it's the p-value is 0.001. So overall, these groupings are significant, even though there is some overlap, as you can see. So we have these distinct color patterns. Did these evolve from convergence? So I made a phylogenetic tree based on 2 genetic loci, ITS 1 and 2, but that's not important. You can't read any of the names, and that's not important either, so I'll just show you. If I look at this species right here, it falls out on that on the tree. That one's right there, and that one's right there. So you can see these are not that closely related to each other, but they look a lot like each other. In other words, they evolved from convergence. And if you look at these little symbols at the end of the tree, green is all over the tree, red is all over the tree, and so on. So it's pretty randomly distributed on the tree. It's not all of the green ones or green designated ones. They're not all in one spot, generally. So yes, these color patterns evolve from convergence. The last one, are color patterns sympatric? Do they co-occur geographically? So if we just look at the general distributions of these species, as I click through here, you can kind of see a pattern is emerging. And all of these, they have that color pattern, and they all live right there. There's a lot of overlap in the distributions of those species. So yes, they co-occur geographically. And I expanded this to include those 67 species and came up with this map. So there's a lot of, you can see from the map, green ones are down there, these ones are in the eastern United States, these ones down here in the western United States. So there's a lot of, there's a strong geographic component to these color patterns. So from this Mullerian mimicry complex in Dasymatilla, there's a lot of potential here. This is just recently described, so we can use this when teaching about natural selection or teaching about mimicry. A lot of research questions that can be asked about that. And with the 67 species, it makes them one of the most diverse mimicry rings that we know about. The butterfly ones in the tropics have dozens of species, but 67 is quite a lot. But we're just getting started. What I mean by that is these 6 species here can easily fit into one of those mimicry rings, and I outlined them so you don't have to work too hard. But these are actually from a different genus of velvet ants. And so when I expanded my search of velvet ant mimicry to include all diurnal velvet ants in North America, I found a lot more species were involved. So in I call this the Texan mimicry ring because it's kind of in Texas. It has now 2 genera in it. These are non-Dasyutila species. The desert mimicry ring was in the desert, and it has 2 genera also involved with that. The Western mimicry ring, I expanded it to include 4 genera and also expanded the range a little bit down into northern Mexico. Eastern mimicry ring, 7 genera, and it's in the east. Madrean mimicry ring, named after the Sierra Madre mountains there, 10 genera, and there are a lot of species involved there. Tropical mimicry ring with 12 genera, it's mostly in the tropics. And then there was 2 new mimicry rings, and I haven't figured out what to name these yet, but we call it the Timula 1 mimicry ring. All of the, from about here down, are all the same genus of wasps, Timula. But then we have all these other genera and species that look like Timula. So then there's also a Timula 2 mimicry ring. Again, I think of some more clever names than Timula 1 and 2, but they look like each other. There's 7 genera involved. The difference, if you're wondering, between Timula 1 and Timula 2, these ones have black heads, these ones have non-black heads. So I wanted to do the same kind of thing with the non-metric multidimensional scaling analysis. I wanted to test the similarity of these mimicry rings statistically and more rigorously. So a colleague of mine who loves to look at velvet ants under the microscope came up with these 54 characters. So he looked at each one of these spots on every one of those species and coded it based on that key. If it was black, it got a 0. If it if it was white, it got a 1, and so on. So we made this big data matrix with all those species. Again, we tested that with the non-metric multidimensional scaling, and there's lots of dots on there. But these dots, if we look at it, the green ones mostly are here, that's those species. Black ones are mostly down there. So there is some clustering. And when I test this with a permanova analysis, it's significant. So these are significantly clustering together with a lot of overlap in some. So when all together, I put this together, and we see that there's more than 300 species involved, 24 genera involved, and 8 distinct mimicry rings. That makes this by far the largest mimicry ring that's ever been described. But what do we do? What do we do with that large mimicry ring? Well, some people have suggested that there's some Batesian mimics of these velvet ants. So this is a jumping spider that I caught in St. George. It looks kind of like these western mimicry ring velvet ants. That's a beetle that lives in the eastern United States. It looks like some of these eastern mimicry rings, or eastern species. So there's some Batesian mimics, relatively harmless things, mimicking the velvet ants. But what about other bugs? Are there any mullerian mimics involved with this mimicry ring? So I have a friend who studies spider wasps, and she suggested that there are spider wasps, specifically in this genus Sorthaspis, that might look like velvet ants. Now, what is a spider wasp? Spider wasps, as the name indicates, kill spiders. Actually, what's pretty cool, what they do is they find a spider, they paralyze it by stinging it, and then they drag it back to a nest. This is a paralyzed spider. That wasp is dragging it by the connection between its head and abdomen, cephalothorax and abdomen. So all spider wasps kill spiders, and a lot of them are specific on the kind of spiders they kill. Sorthaspis specifically attacks trapdoor spiders. They look kind of like tarantulas, but a lot uglier, and they live in these burrows with trapdoors on them. So because the majority of this Sorthaspis's life is spent on the ground looking for these trapdoor spider nests. And also because the Sorthaspis, it finds a trapdoor spider, it stings it, and then it just brings it back down into that nest. It doesn't have to dig a hole like this one will. Most of its life is just spent wandering around looking for these trapdoor spiders. Most of the velvet ant's life is spent wandering around looking for bee nests. So they have a very similar natural history in that they're wandering around on the ground. So I did the same kind of analysis, but I included members of the genus Sorthaspis in with these velvet ant mimicry rings. And so you can see that there's some morphological similarity, that some species look like they fit in the western ring, eastern ring, and so on. Then I also looked at the geographic distribution of the spider wasps down here compared to the velvet ants over here. You can see there's some similarities, right? The eastern mimicry ring ones, these 3, are in the same area as the eastern mimicry ring velvet ants, and so on. There are some big differences though. For example, the western mimicry ring spider wasps, these 2, go a lot farther down into Mexico than the velvet ants do. And then the Mondrain mimicry ring, that species right there, that specimen there, it ranges farther south also. So there's a little bit of differences, but it looks like overall it fits pretty well. I did the same kind of analysis when I measured the morphological characters, and I put the spider wasps in that plot with the velvet ants, and you can see they fit pretty well with the velvet ants. Pretty well. So now, if we look at this mimicry complex, we see 310 species, didn't add that many more species, but from 25 genera and 2 families. So still 8 distinct mimicry rings. What's next? Well, there's a lot of things we can do with this. This is a really cool system and a lot of unanswered questions. But today I'm going to talk specifically about imperfect mimicry and what drives the evolution of imperfect mimicry. So most people that study this study it with flies and bees. These two on the left are flies, and that's a bee on the right. Sometimes a hoverfly, this is a hoverfly, looks a lot like a bee. I think those two look really, really similar. That would be pretty perfect mimic. This is another hoverfly species, and it doesn't really look like that bee. You can say it kind of looks like a bee, but it's not a perfect mimic of that specific bee. Hoverflies and studying imperfect mimicry in hoverflies is difficult, but last year there was a big analysis. I think it was last year, 2012. Yeah, that's last year. Big analysis. They looked at all these hoverflies, and then they compared them against these potential models, bees and wasps. And for some reason, these are, to me, they're upside down. I would put the bees facing up and the hoverflies facing up, but this is how they did it. So they calculated the degree of imperfectness between a hoverfly and any of these models. I call that mimetic fidelity, how well the mimic matches the model. And what they found this is from Penny et al., but what they found is this, and I'll explain it in a second. So if you were this predator, that's a bluebird, which of these flies would you choose if they looked like that? Right or left? If you were hungry.
The one on the right.
The one on the right, because it's big, right? It's a good meal. You could eat 5 of those, or you could eat that one big juicy one. And so what Penny et al. Found is that the bigger bodied flies were more perfect mimics, and the smaller bodied flies were less perfect mimics. And their explanation for this is a relaxed selection explanation. So natural selection, because that is the preferred food source, natural selection will strongly favor the ones that look a lot like a bee. If you look more like a bee, you won't get eaten as often. If you look less like a bee, you're going to get eaten pretty often. So quickly driving the evolution of a more perfect match between a mimic and a model. But if you're a small fly, you're going to get ignored a lot by the predator. And so there's going to be a relaxed selection is the idea here, that the small ones aren't getting eaten as much. So even if they look kind of like a bee, the bird might say, oh, I'm going to ignore that little bee-looking thing and I'm going to eat something bigger. So that's the idea that these researchers found. And I wanted to know if does the same thing happen with velvet ants. So you can see we have some velvet ants that look a lot the same. We have others that don't look that much the same. They're kind of similar, but they're definitely not as perfect mimics as those two are of each other. And now remember the difference between Batesian and Mullerian mimicry. Batesian mimics, there's a harmless one like the flies mimicking a bee that stings. In Mullerian mimics, these are both harmful. And so it's a little different dynamic going on. But before I could test any of the imperfect mimicry hypotheses, like the small body size hypothesis, I had to estimate mimetic fidelity for velvet ants, how well the mimic matches the model. So in this case, the model is that whole mimicry ring, all of the harmful species that live in that area. So what I did is I took a group like this, and I gave them all pieces of paper to rank how well a mimic matches a model. That would be the mimetic fidelity. So if I ask you guys, that is a mimic, how well does it match that model on a scale of 1 to 10? It doesn't have to perfectly match any of them, but how well does that fit in a group? What I actually told my students is, if you're a lizard, and you know all of those will sting you in the mouth, what's the chance you're going to eat that one? How well does it match it? So what would you guys say? On a scale of 1 to 10, with 10 being a perfect match, 1 being a horrible match, how well does that match that group?
Say 10.
You say 10?
I wouldn't eat it.
You say high? You would eat it? I wouldn't even eat it.
Yeah.
Anyone disagree with 10?
No.
When I did this, my students, I think I did this with 150 students or something, they gave it an 8.2 on average. So pretty high mimetic fidelity. I did it with all those different mimicry rings. So we have this one. How well does that one match? What would you guys say?
That's lower.
What number? What number? 7. You say 6.5? 7.5?
It's that the variability between all of them is greater than the variability between that and the average.
And so that's what you kind of have to take into account. If you're a predator looking at this, you know that all those are bad.
Yeah.
And so is it more variable than this, or is it less? It's a difficult problem for students and for predators. My students gave us a 5.8, and I don't disagree with it necessarily. But so I did this with all these different velvet ants, and I measured the size of all these velvet ants. These are projected to look the same size, but in fact that one is about twice as large as that one. So there's a big body size difference. And what I found, does size matter? Not really. And it really depends on which mimicry ring you're looking at. So for example, this one right here, the brown dots or brown triangles, that's the Madrean mimicry ring, which is the very top of that graph, of that figure I keep showing you. So for those, the smaller ones are less perfect mimics than the larger ones. But for others, like these red symbols, which are these ones right here, the smaller ones aren't any less perfect than the bigger ones. So the small body size hypothesis that was developed from the hoverflies doesn't really seem to fit very well with the velvet ants. The smaller ones aren't less perfect mimics. But there are definitely less perfect mimics, so what's driving that? So I kind of looked at it as a whole, I took a step back and looked at it by mimicry ring. So this is the Texan mimicry ring. Those are all pretty perfect mimics of each other. The Madrean mimicry ring is not. So this could be because I'm the one that described these mimicry rings. Maybe I just lumped these together and they shouldn't really go together. But I think that it's actually something different. So I looked at what I'm calling community diversity. This isn't a true, I'll talk about it in a second, but it's not true community diversity. Community diversity would be how many species live in a certain area. But what we find is, and I'll explain it in a second, what we find is that mimetic fidelity is significantly related to community diversity. That's what this line is right there. And so then in each of these box plots is a diversity measure for how these communities. So in other words, the Madrean mimicry ring is in the most diverse community. The tropical mimicry ring is in the least diverse community. The problem with this is we don't really know the community diversity. We don't have the data for it. Not a lot of people collect velvet ants, and when they do, nobody databases the information. So we can't say in Orem, Utah, community diversity is 1, or whatever metric we're using. So what I did is I used this map as a surrogate for community diversity. And because these are mimics of each other, we're not looking at species diversity, we're going to look at morphological diversity. How many different colors of velvet ants do you see in a certain community? If you're a lizard and you live up in Idaho, almost all the velvet ants you see are going to look the same. There's going to be a really low morphological diversity of velvet ants because there's only one mimicry ring that lives up there. If you live in the east, it's similar. But if you live right there in southeastern Arizona, you're going to have some white velvet ants from this blue mimicry ring. You're going to have some orange and yellowish velvet ants from this mimicry ring, black and red ones from this mimicry ring, and then those colorful ones from. So there's a lot of different colors there. And so our hypothesis that we developed based on this, that we're still gathering data to test it, is the community diversity hypothesis. If you're in an area with a lot of morphological diversity, if you're a predator that's there, you're going to learn quickly that all these different colors of velvet ants are bad. And so anything that looks kind of like any of the colors, you're going to avoid. There's not a lot of strong selection pressure to look exactly like any of them. You just have to look kind of like some of them. But if you're up here in northern Idaho, the predators are only going to know the orange ones are bad. And so if you look different than an orange one, you might get sampled. The predator might test the waters and see if you're good. And so what we're working on now is testing this community diversity hypothesis. I want real community data. I want to look at collection sites all across the country and really get numbers for how many different species live there or how many different color morphs live there. Another explanation of imperfect mimicry is called the intermediate mimicry hypothesis. So in this hypothesis, we have a wasp here, a bee here. Both of those are bad. We have this hoverfly here. It looks kind of like the bee, and it looks kind of like the wasp. In this intermediate mimicry hypothesis, that is going to be more imperfect because it's getting benefit by looking like both models. It's intermediate between the models. So if I'm a bird and I see that, some birds might say, yellow jacket, I'm not going to eat it. Other birds might say, honeybee, I'm not going to eat it. Either way, they're not going to eat it, and so it does just fine. So with velvet ants, it's a really good system to test this because we have a lot of somewhat intermediate forms. So we have these white ones in the desert mimicry ring. We have these reddish, orangish ones in the western mimicry ring. Then we have some like this that are, I've classified it in the western mimicry ring, but it has some obvious similarities to those white ones. And it's interesting because this is Dasybutyllus satanis. It's a really big velvet ant. Maybe that's why they call it satanis. I'm not sure. In the northern parts of its range, it goes up into the Central Valley of California, up into St. George. It's orange. It's kind of that color. Still hairy, but it's orangish. As you go farther south, like down by Yuma, Arizona, it's this lighter yellow coloration. So that could indicate, and I'm still working on this, that in the northern parts of its range, it's mimicking these ones. In the southern parts of its range, maybe it's a more intermediate. Maybe it's getting benefit by looking like the white ones. I'm just this semester, I'm collecting data just like we did with the mimetic fidelity for these. And I've just looked at it this week, and it looks like that when I compare this to the western mimicry ring, it gets a mimetic fidelity at about 5. And when I compare it to the desert mimicry ring, it also gets a mimetic fidelity estimation about 5. So at least people think that really looks in between those. What I'm going to do next is measure a bunch of morphological characters, plot that in ordination space, and see if it's also intermediate in ordination space. But I didn't show that data here because I didn't want to throw it together at the last minute and then forget what I was going to say about it. And so I talked pretty fast, but we'll have lots of time for questions, I guess. I have to acknowledge some people. Kevin Williams is he was at Utah State when I was up there as a graduate student. Now he's just starting a job in Florida. He's a velvet ant geek, and I'm sure he would call himself worse, but he's the one that really loves to look at the velvet ants and is the one that measured 52 characters of little velvet ants. Is what his life is. James Pitts is also a velvet ant researcher up at Utah State in Logan. Matt Forrester and Josh Jenner are in Reno, University of Nevada, Reno, and they've been helping with more of the ecological side of this study. And that's what I have for you. And I'll take questions because we have 20 minutes or so. Gotta talk slower next time.
I have a question.
Yeah.
Does the mimicry, do you think that has to do with the outcome of the sampling? Does that make sense? Like if there's two birds sitting together and they eat one and one bird dies because he ate it, you know, that other bird's going to say, I'm staying clear away from everything. But if it just, oh, that was gross, then they might have, you know, the app to test something that looked close to it. Does that make sense? My question, I'm not really...
Which one do you mean is going to test? Is it the one not the one that died?
Yes, okay, so there's a, let's say there's a red velvet ant.
Okay.
Two birds are sitting there, hanging out, you know.
Got
and
it.
one samples it and it dies immediately.
Okay.
I think the other one will learn, well, you're going to die if you eat something that looks like that.
It was only if the
Rather
bird
than
is
just
cognitive
taste.
enough to know, hey, that just ate velvet and it
Okay.
dies, which probably
Well,
isn't the case. But.
Okay, so that it's just,
But they don't die from eating
No,
velvet
they
ants.
probably won't. So there's only been a handful of studies seeing what will even eat these. I'm working on it with a friend. We're trying to feed them to everything they can think of. And so far, nothing eats them. Except the only thing that tried eating a dead one was a juvenile bluebird in Indiana. And so juvenile is important because it's a bluebird that hasn't experienced much in life. And so it tried to eat a dead one. But the other studies that have successfully tried to feed them things, they did big-headed skinks. It's a big lizard that lives in the east. It's called big-headed skink because it has a big head. And they have really powerful jaws. Is it big-headed or is that right? Do you know skink stuff?
I think so, yeah.
Anyway, it has a big, powerful jaw. And these researchers said big-headed skinks eat stuff that's hard. Velvet ants live in the same areas, but I've never seen them when I look at the guts of big-headed skinks. Herpetologists look at guts of lizards. So they tried it a bunch of times, like 20 different times. And out of those, I think it was 27 times, 27 different lizards, 27 different velvet ants. 2 of them were able to eat them. One bit it in half and ate the head and thorax of the velvet ant so it didn't get stung. The other one got stung a bunch of times and just kept trying to eat it. Other ones, they would bite it, get stung, and immediately let it go and never try again. And so sorry, I'm not really answering your question.
No,
but
you're all right.
Things
My
that learn
thing
quickly
is,
to
what
hide.
are they trying to defend themselves? You just
These?
haven't found a predator of the velvet ant.
That's a good
So,
question.
you know what I'm saying? Why is
Yeah.
the diversity, and why would they want to mimic if they really don't have a predator?
Oh, it's a great question, and that's what one of the things that makes this unique is that there's not a clear predator. And so what is driving this, what drove the evolution of this? Why don't they just look
Yeah.
like the nocturnal ones, which are
Mhm.
brown? Except for that one's a nocturnal one, but we'll talk about that later. I don't know. I don't know what's driving it. Do you have idea?
Well, I was going to say sexual selection. You looked at, with these, as you pointed out, the males and the females sometimes are very different colored, and I'm assuming that vision. I mean, they have big eyes too,
Yeah.
right? So they have good vision, and I'm assuming that probably mate choice and things might be part of the story, especially. When you look at your phylogeny, you see this thing is all over the place. I mean, this is a very plastic character that genes are changing colors all over, or genes are being turned on and turned off, and changing the color of these things quite rapidly. I'm not sure, what
That's the only thing I had in my mind is something to eat it. I didn't even think about the.
Yeah, well, the sexual selection one is a good. It's an area that we need to look into. The problem is we don't know almost anything about the mating behavior of these. So the females crawl around. That's all they can do. The males fly around, like a normal wasp would. They go to flowers and drink nectar, fly around looking for a female. I think a lot of it's chemical communication at a certain point. They have to fly around looking for the pheromone in the wind, and then they hone in on where the female will be. The only records we have or observations that have been published of mating is supposed to be really, really fast. The male comes in, lands on the female, and flies away, and it's done. I doubt that's really what happens. I think people just haven't seen it much. Like most bees and wasps, or all bees and wasps, they mate once. So the female emerges as a newly emerged female. The male comes and mates with her, and then he's done, and she saves the sperm for the rest of her life and uses it when she wants. And so it's not easy thing to find is a mating pair. And so it's hard to say if it could be sexual selection. It might be, because I have seen areas where 2 different species, 2 different color forms are active at the same time, and there's males looking for the females. But I'm not sure. But then the geographic component is also confusing too, because if it's more of a female choice, why do it seems like there should be more divergence in a geographic location. Yeah, because if you're in one area and you look like your neighbor, then your neighbor might attract the male when you want to attract the male. So there should be some divergent selection saying, if you're in the same area, you need to look different than me, so my male comes to me. It's oversimplified and anthropogenic.
Yeah, you said they also make these squeaking sounds by rubbing. Might that have any indication for you're talking about, which, do you want to make with me or my neighbor kind of thing,
Yeah.
Are they, because they're solitary, I mean, would they be able to pick up the frequencies of those squeaking, or do you think they'd be able to use that as some kind of a differentiation?
So the squeaking has some components that hopefully next summer, because you have to study live ones for the squeaking, unfortunately, and it's a lot easier to study the dead ones, but I have two questions about the squeaking. One could be that, and I'm not sure, it's pretty loud, but I don't think that it could be heard very far away from, so maybe they could hone in chemically and then hear the squeaking, but because we haven't seen many mating, we don't know if they squeak when they mate. I have seen one pair of nocturnal velvet ants squeaking while they were mating, but I don't know if they were squeaking because I was looking at them with a headlamp, or if they were squeaking because they were mating. But I know they squeak as an anti-predator thing. So another question for me is there mimicry in the sound? Because if the predator is learning, it bites it gets stung in the mouth, it sees the bright colors as it runs away and hears the squeaking, could there be mimicry in the pitch or the sound of the squeaking? And so I need to record a bunch of squeaking velvet ants next summer and put it into some software and analyze it. That's a good question.
How long do they usually live?
That's a good question too. So last month I was in Albuquerque, and there's a natural history museum. There's a spot for kids that have all these stuff to look at. Anyway, they had some velvet ants in there, and I was talking to him about it. And he said they've had them for over a year in this cage in the natural history museum. So in captivity, at least a year. Outside, we don't know. And those are the females. The males probably live a lot less, a lot shorter, probably less than a month. The females probably, we don't know, a couple months probably.
As the adult. you're
As the
talking.
adult, yeah. And we also don't know, we don't know how many eggs a female can lay. So is their life, I mean, how are they distributing their time? Are they looking for nests, or are they parasitizing a bunch of things? And so I don't know. But if they live longer, it would give more time for a lot of stuff to happen. And there's probably different ones that live short and different ones that live long. We don't know any of that, unfortunately. Any other questions? Has anyone been stung by a velvet? So one of the things they call these, since we just have a couple minutes, have you ever heard them called cow killers? So it's often in the East, they'll call these cow killers as the common name. And I don't know why, I don't think anyone really knows why common names become what they are, but the story is that there was a rancher, he saw one of them on the ground, and it's a fuzzy looking ant, so he picked it up and he got stung, and he said, ow, that hurts bad enough to kill a cow. But if you're talking to people and they say something about a cow killer, or if you are bored and you're YouTubing stuff, YouTube cow killer, and you'll hear them squeak. So there's lots of YouTube videos of them squeaking because people put them in a box and videotape it and put it on YouTube. I don't know. That's what people do. So, yeah.
You know, they use the spiders mostly for raising their young, if you will.
The spider wasps
Foods,
do, but
yeah.
these ones don't.
Okay, that was one. Do they eat nectar, or what is it?
So the velvet ants use bees and wasps to feed their young. We observe some nocturnal ones crawling up on flowers and drinking nectar. So that happens sometimes. But they also, we think, they crawl into a nest of a bee and they parasitize the larva when it's big. So right before it pupates. And so they actually wound it and then drink the blood as a liquid and protein source. So they're fairly mean.
Why are the seeds so large or so long?
Oh, so that's another thing I'm working on, actually. So he asked why the CD is so long. It's why are some really hairy, like the white ones that I showed, and why are some less hairy? And I think there's a couple things that it could be, but I think it might be a thermoregulation thing. In the East, where there's more cover than there is in the West, their hairs are almost always short. In the West, where it's often more open and arid, the hairs are really long. And especially in the hottest part of the West, in the Sonoran, Mojave deserts, you get the white ones. And so I think that the long CD might be shading them, and then the lighter colors might be doing solar reflectance. I'm working with, I can't remember his name now, but at the University of Utah, he has a really fancy $40,000 camera. And it measures the temperature as you take the pictures, and you can do videos of it. So I did it with some live ones this summer, and I'm going to take a bunch of preserved specimens and put them under a light and measure the temperature and see how fast it races. So then we can compare color, how that affects the body temperature, and the CD length and how that affects it. Because as they're crawling around the ground, the surface of the desert gets really, really hot. And if it's too hot, they can't look around for nests of bees. And in fact, they're most active when it's summertime. They're most active before about 10 o'clock in the morning, and then in the evening right before it gets dark, because that's when it cools off enough to walk around the sand or the dirt. So I think that probably the CD length probably has something to do with that. Some people suggest camouflage also, but that's another story. Any other questions? Yeah.
Just generally, I guess, what kind of puzzles do you see that investigating this is kind of being able to lead to?
Yeah, so it's a good question. So we know a lot about, well, we think we know a lot about some mimicry systems. The butterflies that I showed at the start, the heliconius butterflies, we have genomic data for them, and we can search the genome and look. They've identified the genes that are involved with the different wing patterns, and they see a lot of sharing of genes between closely related species. So we understand that system pretty well. There's also a lot of ecological work done in that system with different places they fly in the canopy, and so we kind of understand that. But that's a tropical system, and so people have suggested for a long time, and I didn't mention it here, that mimicry is more common in the tropics. Those examples I gave, there's tropical butterflies, there's tropical frogs, other tropical butterflies, a lot of tropical mimicry things. And that's been suggested because there's a lot of predators in the tropics. More diverse predator community and maybe longer lived things. But there's hardly any mimicry complexes described from the temperate areas. And so what this can do is give us a different kind of system to ask the same questions that have been asked in the butterflies. What's the ecological components driving mimicry in this? What are the genetic components driving mimicry in this? So it'll just help us get a better understanding of mimicry. Then why do we care about mimicry? Well, mimicry is a really good example of natural selection. It's a pretty straightforward thing. If you look like this other thing, you don't get eaten. If you look like your own something, you get eaten. So it's a pretty good way to explain natural selection. So hopefully we can develop this system into a long-term study system to look at what influences mimicry, especially Eulerian mimicry, which there is not a lot of work being done on currently. But that didn't really answer the question. There's lots of puzzles. Pretty much anything you want to know about them, we don't know. So, looks like you have another question though.
Oh, I'm trying to form something. In my
Okay.
head. So, wait, there's the Muellerian, and then what's the other one?
Batesian. So Batesian is when there's harmless and harmful.
Yeah, okay, so, the Batesian one seems to lend, I don't know, a little argument against evolutionary theories like handicap principles, like I mean, basically you're trying to find a new energy niche by mimicking something so that you don't have to dedicate resources into building a stinger or poisons. But then you're actually a weaker version.
So you're
not as fit in certain ways.
So you think about it from a natural selection standpoint. If you're a fly and you live in areas where bees live, which is everywhere, if you happen to look a little bit more like a bee, you will probably pass on your genes better because you're not going to get eaten as quickly. Does that make sense?
Yeah.
And so then your offspring that look more like bees will also pass on their genes. And so you get this slide towards flies that look like bees. And it does weaken the system overall because if a bird's first experience is eating a fly that looks like a bee and saying, wow, that tastes good,
And so easy to find.
then it might say, these orange and yellow things are really good. And it might do it again and happen to get a fly and say, I think I found my role in life, eating these orange and yellow things. Then it eats a bee and gets stung and it has to relearn that. So it's Batesian mimicry is detrimental or it's a negative effect on the stinging thing.
Yeah.
But it's good for the non-stinging thing. So it's a little bit of an interplay with how do you train the predators effectively. And so if there's too many flies in the area, more bees will get eaten. If there's too many bees in the area, then no flies get eaten. But I don't think I'm answering your question.
No, you are. It's inverse frequency dependent selection. So it's true that early on in natural selection, it is very beneficial to the mimic, but then it becomes less and less beneficial to the mimic. So it's not an argument against natural selection at all. It's actually what we'd expect to happen as the mimic becomes more frequent.
And so that's also part of the imperfect mimicry stuff, because you can look at the diversity of the mimic or the model, and if there's more mimics, then there could be less perfect mimics. If there's more models, they would be more perfect. But it gets really complicated. And that's why Muellerian mimicry is kind of nice, because it's a little different than Batesian. Because people often try to say which one's the model in Muellerian mimicry. Some people say, well, it's the most abundant one. If one species is really abundant in the landscape, then things are going to try to look like it. But really, they're all the model for each other, because as soon as they start looking like each other, they're both benefiting from it. So I don't know if there wouldn't be a frequency-dependent component to Muellerian. Positive. Oh yeah. So as you get more in the area. And so that's why, the spider wasp stuff is just something we're just barely working on, because with such a big mimicry ring in velvet ants and with no apparent predators, a lot of things should be trying to look like them. And so we have the spider wasps that kind of look like them. There's a bunch of jumping spiders that kind of look like them. And some people say jumping spiders are harmful too because they bite, but a lizard will eat a jumping spider a lot more easily than it will eat a velvet ant. So there's lots of other stuff that kind of looks like them. But so then it kind of gives us a bigger system to ask a lot of questions with. And also the good thing is, so if you're studying butterflies in the tropics, it's hard to have a big cage with butterflies with birds trying to eat them. It just is difficult. But with velvet ants, you can keep them in a terrarium for up to a year. And you can keep lizards in a terrarium pretty easily too. So we can do a lot of feeding trials to really get at what's happening here. How fast do lizards get trained? Or I tried to feed one to my praying mantis last week, and it didn't work. But do invertebrate predators also react the same way? Long-lived predators will probably be trained more effectively than short-term ones. Another question that's interesting about it, so I'm going to go back here so you don't have to look at me riding velvet anymore. But these black ones right here, black and red ones, the Texan mimicry ring, some of these, like that specimen there, super small, probably 5 millimeters, pretty small. This one right here, though, is about 1 inch long. So it's one of the puzzles that, as you asked. So why would, or how would they benefit each other? A predator that's going to attack the inch-long velvet ant is probably not going to attack the 5 millimeter long velvet ant. Sorry to mix metric and non-metric. An inch, 2.54 centimeters versus 5 millimeters. But so it's an interesting question, because a predator that's eating the little one probably is never going to even think about eating that big one. And so then how are they benefiting from looking the same? Because they need to be training a predator not to eat either of them. And I'm not sure what it is. It could be that juvenile lizards, if lizards are the predators, juvenile lizards are little compared to adult lizards. So maybe the juveniles are learning from these guys, and then as adults, they're also avoiding those guys. If a lizard remembers that long. I'm not sure if a lizard does. Another interesting question, so there's one other mimicry ring that's just kind of getting developed. Well, I mean, it has evolved, but it's just getting described. And that's with bumblebees. Heather Hines is at Penn State. She works on bumblebees and mimicry, and she has hundreds of species also. I'm still winning with 300 plus species, she says. But there's a lot of bumblebee species involved with Mullerian mimicry. Bumblebees sting and they look like each other. The difference is in the bumblebee mimicry, there's like 30 different mimicry complexes. I have 6. And so with hers, it'll be, there's a mountain range in California and there's 2 species that look like each other. Or you go south of the mountain, there's 4 species that look like each other. But mine, I have however many, 15 species that look like each other from central Colorado all the way to the coast. From 8,000 feet in elevation, you see this one. And down to sea level, you see this one. So there's something going on maintaining these color patterns across big geographic areas, ecologically very different habitats, but they still look like each other. There's another component that I am now out of time, but I want to look at the history of this, because there's a fossil of Dasymitilla from Dominican amber. So Dominican amber is right there somewhere. It's a male, which is tricky because they don't look like the females necessarily, but it makes it so we can estimate the age of the phylogeny. If I zoom way back into the phylogeny. Animations makes it zooming hard. We can estimate the age of all these diversification events. And so that's something I'll do. I need to make a bigger phylogeny. This is 67 species and there's 150 Dasymitilla species. So when I make a bigger phylogeny, I can estimate the age of this. And one of the patterns I see on here is that these blue dots are often next to an orange triangle or yellow triangle. So that means the desert mimicry ring is often sister species. So desert mimicry ring is often sister species to a western mimicry ring species. That could be some of that thermal regulation. The deserts aren't very old in North America, geologically speaking. It depends on how old you think old is. Several million years. But velvet ants are older than that. So this starting about right there is estimated to be about 20 million years old. And so it could be that when the desert started to dry out in the Miocene, maybe that encouraged these white ones to evolve as a thermal regulation thing. So I'm going to be able to investigate that more thoroughly as I get some more data. Yeah,
One last question. So you've done this all on the females.
yeah. I actually took that part of my talk out because I thought I'd go over, and apparently I didn't.
Any indication that the males are, consistent and contiguous with the?
Yeah, so we've actually done a little, so one of the problems with that is that we only know, so males and females don't look like each other. So it's really hard to tell what male goes with what female. And only about like 18% of the males and females have been connected. So in other words, we only know what male goes with 18% of the females, roughly. So most of these, we don't know what the male is for them. When they describe them, the male is described as a separate species, and so it's hard to say. But we're in the process of that. We're using some molecular techniques to say this male goes with this female, and we can link that up. And we've done it some. And so the other interesting part of that is males don't sting. They squeak, and they're aposematic, and they have hard cuticles, but they don't sting. So they would be Batesian mimics of the females, which is interesting too. But for example, this species right here, the white fuzzy one, its male, let me go back to this. Oh, where's my map? Sorry, bear with me. Animation. Here we go. This white one right there, when it lives here, its male looks like this with wings. When this white one right here lives right there, its male looks like this one with wings. So it looks like the males are sometimes participating in the mimicry complexes, but not always the same mimicry complex as the female. When we map it on the phylogeny, it often looks like the males are phenotypically more similar to their relatives than the females are. So in other words, the selection pressure is on the females to look different. And the males look like their brother. But with only a few, with all the holes in what male goes with what female is hard. But we do have a lot of that data in progress.
I'm going to ask one more question. So if we were down in southern Utah and we see, how would we maybe look for these? Like they'd be on the ground, but
yeah. So it's often, it's tricky. Sometimes I'll go and look for them and find nothing. Sometimes I'll find 15 of them. And so the way I find them most often in sandy areas, not in the open sand dunes where the four-wheelers are riding, but in the margins of the sand dunes where the four-wheelers are killing the vegetation. So you walk along the sandy areas, often before it gets too hot. The little harvester ants, those red ants that people call a lot of different things out in the desert, if those are crawling around, then it's probably cool enough for velvet ants to be crawling around. But you just wander around the desert and you see them crawling. And it's what we don't know any of the phenology, so the timing of when they're active. I'm working on developing a database of specimens so I can look at that. Looks like late summer is the best to find them. You walk around the sand dunes and you see them crawling around and you catch them gingerly. And so that's one thing I want to do is just follow one around for a while. The problem is when it sees me, it runs away. So I have to try to follow it from a distance so I can see what it does. I've tried it at night. I've done a lot of work on the nocturnal velvet ants, and that's hard because you can only find them with the headlamp. And then once you see them with the headlamp, they know you've seen them.
It's night vision.
Night vision
No.
goggles.
Well, I've tried it with, so a lot of bees and wasps can't see red, red color, so I have my headlamp has a red setting. And I've tried that, but I can't tell if they might also be feeling my footsteps in the sand and running away. It's a tricky business, but it's pretty fun. So I have one in my office right now that I've been trying stuff with alive in my office. I've mostly been trying to feed it to stuff, but so I have a tarantula and a praying mantis to try to feed it to. Is this still on?
Yeah.
Yeah.
Thank you.